Okay, thank you, Dan. It's really a pleasure to be here today. I have to say that putting together the genome has been a tremendously exciting event for me professionally, something very different coming from an algal background, but then moving into seagrasses, which I consider the higher algae. And we're finally here today. And as you probably all know, and I'm going to do this a little bit backwards, is that putting a genome together takes really a big team effort. And that's certainly been the case here with some 35 collaborators in 17 institutions, basically over the whole world. So before I get started, I want to thank all of my collaborators, in particular Pierre Rouzet and Yves van der Peer from the Vlaams Institute for Biotechnology in the University of Ghent. And of course, none of this would have happened without JGI's long-term support. And so we're finally there. And last month, we came out with a paper in Nature, and we even made the cover, which we were very, very excited about. So have a look. I'm not used to this. OK. So let me get us started with why are we interested in seagrasses and why Zoster marina in particular. And I think one of the first aspects of all this is the phylogenetic position. There's some 260,000 terrestrial angiosperms, and among those about 4,500 aquatic or freshwater species. But there are only 60 truly marine species. And if you look on the left-hand part with the monocot genomes that are there, many people think this is just another grass. You know, we don't really need that. But it's not a grass at all in the sense of the Poaceae. It's a basal monocot in the Elismatales. Okay, the closest relative is the duckweed, which has also been recently sequenced. Now, the reason why it's back to the sea is that moving from a terrestrial environment or even a freshwater environment back into fully marine conditions is not a trivial exercise. And when I see things like 140 millimolar salt in our previous talk, when you're in full saline seawater, I don't know what that molarity is off the top of my head, but it is extremely, extremely salty. So you're going to have to think about how you're going to do that. And that's also not an easy thing. So as it turns out, seagrasses they're not a monophyletic group. They're polyphyletic, having returned through 3 separate lineages. What you're seeing there is a classic RBCL tree based on a really good taxon representation. And among those, we have Zoster marina is one of them. And this has only happened 3 times, as far as we know. And that's basically analogous to terrestrial mammals going back to the sea. So they're kind of like whales in that sense. They've done some really interesting things. So we're interested in exploring the genome on what kinds of gains and losses do you expect, what sorts of adaptations have allowed this big transition. Now, the global distribution of seagrasses is everywhere except Antarctica. The highest diversity, of course, as always, is in the tropics, but Zoster marina is the green band across the top and is the dominant seagrass, sometimes the only seagrass, throughout the northern hemisphere. We've done a lot of phylogeographic studies, using older technologies. And over the last 6 years, we've developed a Zoster experimental network called Zen with some 50 member labs throughout the northern hemisphere to look at a lot of community ecology types of questions. Now, the importance of seagrasses, I think, is something that people also tend to underestimate, that they're extremely important in ecosystem function and services. There's a huge literature out there in experimental community ecology about this. In terms of diversity, a lot of pop-gen type of work. But their ecological importance really is that they're a foundation species like forests, like underwater forests, that create a unique ecosystem that's on the same leagues of productivity as coral reefs. They provide habitat, shelter, food source, nursery functions for many fisheries, and coastal protection through erosion by creating baffling effects in soft sediments. They also contribute to water clarity, and the most important one from JGI's perspective in the DOE mission is carbon sequestration. And people, I think, have not really appreciated just how much that really is. And there have been a couple of influential papers published over the last few years, shown here, of how they do that. And if you go to the blue graph down in the lower left-hand corner, you can see that the sequestration rates in absolute amounts for salt marshes, mangroves, and seagrasses as a coastal vegetational type is hugely greater than temperate tropical forests, terrestrial forests, which people tend to, plant a tree, help your carbon footprint. I would say plant a seagrass, help your carbon footprint. But they need to be saved, and it's just huge. And part of that is because the aerial coverage of seagrasses is really tremendous, especially in the tropics, where the water is very clear and they can go quite deep. Having said all that, and putting them there, is also that there is a major global crisis in the loss of seagrasses. There's a lot of anthropogenic effects in coastal areas, coastal development, fisheries, and so on. And there have been a number of pleas over the last several years to pay attention to this system, because aside from the loss of all these ecosystem services, at the same time, one of those is certainly the carbon angle. Now, the seagrass research community, I have to say, is quite a well-established one over the years. There are some 12,000 articles out there. In the sort of genetics, genomics context, it's much smaller, and we're just moving into that direction now. And most of the research community, the really high-powered research committee, are people who do population ecology, community ecology, very much an ecosystem focus, management and restoration. Those are the biggies. And so bringing in a genetic genomic component into the research community has been the task of a small group of us trying to move this forward. I would estimate there are about 300 labs that work in some aspect of seagrasses, and about 20 that are in the genetics genomics area. We're very interested in metasystems, seagrass microbiomes. I'll come back to that a little bit later. And I'm trying to build more of an interest in the monocot community. Seagrasses often tend to fall between the wall and the ship, as we say. Nobody wants to work offshore. They consider that marine, and then the marine people consider them not really marine, but semi-terrestrial, and so there you are. So if you're a monocot biologist and in any aspect, then I want to talk to you. Okay, we need help. All right, so just some genome highlights on what we did. This is a combination of different kinds of sequencing. I was unfortunate enough to begin the project just at the moment that they went from Sanger to 454 and on to Illumina. So it got to be quite a challenging exercise in making libraries and figuring out what's what, but prevailed in the end. We also got some phosmid ends on it, and so just briefly, we have about a 48x coverage of the genome. It's available, of course, at NCBI. The summary statistics are shown here. It's about a 200 MB genome, which at the time that I submitted it to the sequencing program, it was considered a big genome. And now we see that it's not so big after all. Basically, the genome's in about 2,200 scaffolds, and we've got an L50 of around, say, close to 500 kb. And the largest scaffolds are in the range of 5 to 10 million, and we have, quite a few of those. So it's a good start. We're working on a PacBio now to try to tighten that up a little bit. What is interesting, we've got about 20,000 genes, so that's pretty typical. In looking at losses and gains of domains, there's some interesting points there that I don't have time to go into. And then what's most interesting are the transposable elements. We've got a lot of those. They are all over, and the result of that is that if we look here at the 10 largest scaffolds in there, you can see that we've got a lot of gene-dense islands and then a lot of transposable elements in between. We've got a few people who are interested in this in terms of the evolution of the genome architecture, and also people who work with microRNAs in trying to understand the relationships of the role of these transposable elements and their regulation with microRNAs in helping to help the genome evolve more quickly. And here, if we compare with Spirodella, which is the nearest one, you can see that the repeats are much, much more extensive in Zostra with a gypsy dominance, which are typically young and associated with gained genes. So we're pretty sure that these TEs have played a significant role in adaptation to the marine environment. You know, that's what it certainly looks like. And there are a few people now who are interested in pursuing this particular aspect, especially at the University of Ghent, in the evolution of this genome itself. The other thing that we have discovered is an ancient whole genome duplication. And I won't go in again to the details of these, but I'm happy to talk to anybody about it later today if you'd like to. But this is pretty common in plants, as you know. And we think in this particular case, it occurred somewhere around 67 million years ago using KS plots and doing particularly types of modeling to detect it. And you can see also in the bottom panel that many of the monocot lineages have undergone a whole genome duplication in this time frame, which is correlated with the KT boundary. And where there was a huge amount of new ecological opportunity created, and plants, usually when they sense that, however they sense that, tend to get busy and start doing genome duplications. The Zostra genome is not polyploid. It's a 12 chromosome setup, but still interesting. Okay, so moving on to some of the adaptations that we have been looking for in this first pass of the genome is a kind of the gee whiz kind of things. Lots more work to be done. But one of the first things, in case you didn't know it, that seagrasses don't have stomata. And you can see that here in the picture on the leaf here of Zostra as compared with a rice or a Arabidopsis leaf. Now, one could say that, yeah, well, big deal. The genes are still there. They're just not developmentally expressed, which happens to be the case in many aquatic plants, is definitely the case in Spirodella, has really big stomata. But we started to look more into the surface of the cell, the epidermal layers, the cell wall, and down into the plasma membrane, because it's how do you live in salt water? How do you regulate your water balance? So it's kind of the opposite from the previous talk in which there was drought. Here we've got super amount of water and loads of salt. And what we see in doing all this is quite an interesting repertoire of different polysaccharides. And many of these interact with sulfated polysaccharides that are found in the cell wall. And some of these compounds are unique to seagrass and very characteristic of the macrophytic algae, which I like very much. That appeals to me. Now, the stomatal genes have basically disappeared. What you're seeing here are the main stomatal differentiation pathways. And anything that's in red is gone. It's just not there. And so what this tells us is that Zostra is definitely bound to the sea. It's not going back to land anytime soon, unless it re-engineers itself or reshuffles its genes in some particular way to allow that to happen. Now, following along on that line, we also see that the cell walls are macroalgae-like. Now, this has been known from biochemical studies for many years, but it's nice now to have the genome aspects of it, of looking at these different kinds of particular sulfated polysaccharides and specialized celluloses, sulfyltransferases and so on. And these have been looked at by colleagues of ours who are algal cell wall specialists mainly in Roscoff and Brittany. And this is an area that I think is wide open for people who are interested in understanding perhaps some ways that how do you deal with these kinds of ion homeostasis and big floods of salt in water in odd ways. So this is something that needs a little bit more exploration, to be sure. Now, there's photosynthesis and photosensing. Many seagrass people are interested in this. Living in water presents some different photosynthetic challenges, and living in seawater even more photosynthetic challenges, because not only do you have attenuation of particular light waves, but also quantitatively less as you go deeper. And so you're going to need different kinds of photosynthetic sensing, light harvesting pigments, and so on. And at the same time, we see that our basic photosynthesis genes are pretty standard, but we've lost UV protection genes. If you're underwater, most of the time you don't need that. If you have a water film over you, you don't need those, so sunglasses are not necessary. An absence of phytochromes associated with red and far red sensing, and expansion of various light harvesting complexes, and especially the LHCA, LHCb in there. And again, we've always been comparing to, well, what do we know from Spirodella, which is the only other really aquatic plant. And even there, it's not really a submerged plant. Most of the plant body is on the surface of the water and then hangs down, so we don't really know how all of that works. But I think that if you look at zoster, this is also a very fruitful area to look more at photosynthetic processes. And then we went on and started looking at ethylene and other volatiles. And this is, of course, consistent with the loss of stomata, because that's certainly where they generally are going to be emitted into an airborne sort of communication or attraction, largely associated with pollination. But we don't have that kind of pollination underwater. There are no insects underwater. There are some other kinds of crustaceans and things that have been hypothesized to be involved with pollination, but that's not actually been proven. So it's a different sort of sensing, a different sort of plant defense is apparently involved. We've started to look at some of the Ross genes and things like that, and we see that things are generally very much less as compared with other known plant genomes. Here again, in the ethylene pathway, the reds are the missing parts of that. And if we take it a little bit more, we've also looked at terpenoids. That just happened because the eucalyptus genome had just come out, and zoster has only 2 terpenoid genes as compared with eucalyptus with 113 as the champion. And Spirodella, we don't know. We weren't able to determine that. But again, whatever's happening in terms of plant defense in this particular regard, it looks like it's just less. That doesn't mean that there aren't a lot of pathogens out there and a lot of things organisms that have to be dealt with, but it does mean that they're different, and we really don't know about what those things are. And this extends also to defense-associated NBS genes. Zoster has 44, and as you can see, in comparison with others, including Spirodella, it's just much, much less, okay? So we've got to figure out what these pathways and compounds are that are involved, and I think it's a very basic question, but I think a very fascinating one. Okay, so that's just a kind of little highlights there, but clearly the transition into this type of a habitat has required a lot of back adaptations and rearrangements in the genome, and we're just beginning to explore those. If you have your pet area that you would like to explore, let me know. So I guess the question is where do we go from here? It's been a thrill to get to this part, but now what's next? And just a few things that are in progress right now are improvement of the genome itself using some PacBio long reads, looking at epigenetic modification across a latitudinal gradient. We think, based on some transcriptomic studies and other information that we have, that we're going to find that a lot of the climate change adaptability, temperature, ocean acidification, and that sort of thing are probably plastic and not hardwired in the genome at the DNA level. And so we've got a number of different labs working on that. And then we also have, there's a seagrass microbiomes project that is connected through UC Davis with Jonathan Eisen's lab. and in cooperation with the Zen Consortium that basically has geographic sampling from the total range. So that's going on. We're finding some really interesting things there. And then finally, community genomics, which is now that we have a Zostra genome and we have stickleback genomes, and sticklebacks play a big role in many temperate Zostra marina communities. So we're looking at some ecoevolutionary dynamics. The community ecology crowd is extremely excited about that. And then various people are looking and trying to validate some of our pathways, doing some good old-fashioned physiology and experimentation, which we need a lot. And so that brings us back to these basics of osmoregulation, plant defense, photosynthesis, and of course the major macronutrient pathways. Okay, so challenges and opportunities, where we want to go, and we are maybe going to tap into a letter of intent here to JGI, is looking at population and landscape genomics. That's really where the technical horsepower is within the consortium for this kind of thing. So we're interested in a lot of resequencing of Zostra marina, setting up a major big sets of SNP panels and haplotypes to explore different kinds of population level processes, selection being one of them. So that's coupling that, again, with Zen and the microbiome work and trying to expand that. And getting even more grandiose, we would really like to have more genomes. It's the old story, you know, we want comparative data. You know, what do you do with one? And so we would like to be able to expand that seagrass sequencing to picking out at least 2 species from each of the 3 main lineages, and picking 2 freshwater relatives, and picking those up as de novo genome sequences. And that's really important because we'd also like, eventually, this is very grandiose now, we want to look at tropical seagrasses and tropical microbiomes as compared with the temperate ones. No one has ever done this type of work, so that's wide, wide open. And of course, we always need hardcore plant physiology and molecular biology people who would be interested in working on this plant. So join us, talk to me. I can also say that I'm on the verge of retirement, so my motivations are to leverage this genome and move it forward and get as many people in the world working on it and see where we can go, if that's of help in understanding more about salinity and how crops can deal with more saline water. You know, that's one aspect of it, but also just understanding the evolution of a higher plant that has returned to the sea. So I will call it quits here and open it up for questions. Thank you again very much.
Thank you very much, Jeanine. Questions? First.
Thank you so much. I love seagrass.
Oh, good.
They're like a rebel, so I like that spirit.
They are rebels.
So is this species also, does it also clonally propagate like,
yes, it
like
does.
Posteria? So do you have areas in the world where you can see really old, like?
Yes.
And my other question is, what is the generation time from seed to seed and how easy to cross?
The generation time from seed to seed is probably around a year. I don't know how much you could, you know, that's not too bad. If you have access to mesocosms, which they do up at UC Davis, you can do some basic genetics. So I think certainly doing F1 crosses and back crosses, that's doable. But to really do classical genetics or EQTLs and things like that, my personal view is that I think that, yes, it's theoretically possible, but I really don't know anybody who has the resources to do that. So that's a limitation.
And how old are these colonies?
The clones? We have estimated some for Zostra that are on the order of 1,000 years. And in some of the other seagrasses, notably Posidonia in the Mediterranean, they have a peculiar type of mat, and they've done dating there that suggests that these beds are like 12,000 years old. So in a way, you know, we've got immortality here. They're pretty amazing. Yeah.
Chris?
Thanks, Jeanine. That was great. I didn't see much evidence for centromeric regions in the genome. Have you looked for particular repetitive elements that are associated with centromeres? And do you have telomeres also?
Yes, we do, and we do. And we've identified the telomere genes. Okay. Yeah.
And how about the centromeres? It doesn't look like you've got anything obvious.
No, we don't. No.
Other questions? I have one. You mentioned in the adaptation, you said, on one of your slides, you said the adaptation may have required rearrangements of the genome. Mm-hmm. What's your evidence for a mechanistic link between the rearrangements and adaptation?
Correlation. We don't have any cause and effect kinds of things. It's more maybe wishful thinking at a certain point, but correlation is the first step. And what we do see in looking at domain analysis is that there's a huge number of gains and losses in there that are associated with particular protein families that we think are relevant for adaptation to the sea. But
so changes in protein architecture.
yes, major changes in protein architecture. And they found some interesting microRNAs that appear to be unique or very ancient ones. And yeah, I can't really say very much more because it's not my specialty area, but they must be there.
Thanks.
Another question?
Hi, I really liked your talk, and just want to clarify, how much of the 200 megabase genome did you finish in your project? Because I saw still a lot of content, so I didn't catch how many megabase did you have in all the contigs?
In the contigs only, or the whole thing?
Well, the whole thing.
The 203 MB. We think the original estimates for the genome size were larger than that. And so we're probably still missing, I don't know, 30% maybe, something like that. We don't know.
So, yeah, if you're missing 30%, do you think that you can call, like, because I'm really curious about the stome, developmental pathways, where you find evidence that is completely deleted. So in those cases, if you're talking about single genes, would it be presumptuous?
Well, I don't think it is in this. We've had that critique from reviewers on the paper that absence of evidence is an evidence of absence. That old argument. So showing that something's not there is much harder. And the way that they handled that was doing a series of different kinds of blast Ns, blast Ps, looking then at phylogenetic trees, coming back. And the person who did those annotations is pretty much is Pierre Rouzet, and he's a pretty hardcore annotator. So yes, you can always say that maybe that bit that we don't have is where they all are. That is possible. I can't say anything more about it than that. But we took care when we say that it's not there, that it was not just because we missed it or something like that.
Quick question: did you also look into the methylome?
The methylome is being sequenced now.
Okay.
So we don't know yet.
That might answer what Chris was asking about.
It might for
Chris's
for the
point.
Centromere. Yeah.
Thank you.
Yeah.
Okay, Jeanine, thank you so much.
Okay. Thank you.